Our next presenter is Thien Nhan TRAM, National University of Singapore, for breaking the wall of resistant bacteria.
Good afternoon, everybody. In nature, trap and kill tactics using nets is widely employed by many species for catching prey, as seen with spiders, or for self-defense against pathogens, as seen with plant roots, or really small organisms like amoeba. Little do we know that our body also adopts this strategy to deal with disease-causing bacteria. Nets-like structures, known as neutrophil extracellular traps, are released by the white blood cells in response to the presence of pathogens. These tiny nets immobilize microbes and prevent them from spreading so that all the components of our immune defense system can arrive at the site and deal with the threats. Unfortunately, some bacteria can avoid this fate by either cutting up the fibers or prevent their formation in the first place. To worsen the matter, some bacteria have evolved to survive the attacks from not just our immune system, but also from many antibiotics, the very drugs developed to help our body fight off infectious disease. In fact, antibiotic resistance is a serious global health issue. The annual mortality rate resulting from it has been projected to escalate in the next few decades to reach roughly 10 million in 2050. To put that number into perspective, 10 million almost doubles the current Singapore population. In order to deal with this emerging threat, our lab has drawn inspirations from the trap and kill tactics in nature and designed self-assembling peptides for anti-infective purpose. A peptide molecule can be thought of as a really small fragment of a protein. After being introduced into our body, the peptides will be patrolling our bloodstream in the form of individual standalone units. These are constantly on the lookout for foreign bacterial cells. At the moment our peptide comes across any of these microbial invaders, they will start stacking onto each other and assemble into long and thin fibers that can cross-link into a net, which can then entrap the bacteria. Whereas the neutrophil net that I mentioned before can only trap, our peptide net can also kill. In fact, our peptide, the cherry on the cake for this project is that our peptide is also effective against those pathogens that have grown resistant against last resort antibiotics. With our peptide nanonet construct, fishing for resistant pathogens has never been easier. And while we are at it, let's punch some holes through the walls of these stubborn bacteria. Thank you.
Questions?
Are they specific to certain types of bacteria, or are they universal for all types of bacteria?
They are specific to gram-negative bacteria.
Thank you, Mr. Thien.